My pleasure to introduce a friend and colleague, Julien Ayroles. So Julien did his undergraduate in France and then went on to do a master's at University of Illinois, Urbana-Champaign in animal behavior. No, I'm assuming it was Drosophila. Sure, okay, it was flies. Great. Then he went on to do a PhD at NC State, where he was part of the group that developed the Drosophila genetic reference panel lines, the DGRP lines, that many Drosophila geneticists use today. Foundation for many, experiments trying to understand genotypic effects on many different phenotypes. After that, he was a Harvard Junior Fellow at Harvard, of course, and then an assistant professor now at Princeton University. Julien's work is really trying to understand how genes and environment interact to impact phenotypic outcomes that are relevant to the organism, things like reproduction, health, and survival. He does this across two different systems primarily. So he has a fly lab where he studies Drosophila, where we control the environment and manipulate genotypes to explicitly look at gene-environment interactions in really controlled settings. And he also studies the Turkana population in Kenya, northern Kenya, where which is what he's going to be speaking about today a bit, where he's studying gene-environment interactions and evolutionary mismatches in this population in urban and more across the urban-rural gradient of the Turkana people. Julien's won a number of awards. I'll just mention a couple. First is he just had his NIH MIRA, the Maximizing Investigator Research Award, renewed, and he was also a Kavli Fellow. He's got a lot of grants, and I'm also very fortunate enough that I've recently been able to collaborate with Julien and got to spend time with him in Kenya earlier this year with the Turkana population. So I'm very excited to work with Julien on this project, but also I'm really, one of his talents is that he's an excellent safari guide, and I did not know that until I was out there. And I got to check something off of my bucket list, which was seeing an aardwolf, which I don't think I will ever see again. And Julien was the one who found it and was able to lead me to it. So my endless gratitude for that, Julien. And with that little anecdote, I'm gonna let Julien take the stage and tell us about his work in with the Turkana population.
Thank you so much. Yep. Perfect. Thank you for having me. Fun to be here. I've not been in Arizona for some time. I used to do field work in Arizona near Flagstaff many years ago, so it's good to be back. I also have this French thing going, and I'll speak very fast, and so it's totally okay for you to interrupt me during the talk. If you have questions, clarification, please do. This is a small group, informal, so please don't hesitate. So today we'll talk about evolution mismatch and G/E in different contexts. But more broadly speaking, my lab is interested in understanding differences between individuals, what drive differences between individuals. And many of us, this is a common thread between many biologists. Some of us may care about that question because we want to predict phenotype from genotype. Others interested in molecular biology underpinning of this variation. And also this is a stuff of evolution, right? Like Dick Lewinton would say. So this is sort of like how we approach this question. And our thinking has been, I think as a field, has evolved quite a bit about what drives this variation. And that's probably what we'll talk about today. So no big reveal, right? Differences between individuals are driven by genetic differences between us. We also experience different environment. We grew up in different contexts. We eat different food. So of course, there's some environmental effect. But what I'm most interested in are instances where the phenotype of a given genotype is a function of its environment or genotype-by-environment interaction, right? So in this case, whether you're in environment 1 or 2, if you have the AA genotype, makes no difference. But if you have a TT genotype, which environment you're in makes a big difference. And so this is sort of what I'll be talking about today quite a bit. Another way to think about this, which is part of how I got into this subfield, this is here I'm plotting, this is a cartoon, not real data, but like I'm plotting the distribution of a phenotype of interest, maybe BMI, in a bunch of individuals that are in their happy space. Maybe it's like a bunch of people in southern Italy, eating their wonderful food. And I'm going to take these same people from that same population. I'm going to put them on a McDonald's diet, something like that, away from their happy space. And this is what happens. The first thing you notice is that there is a fairly dramatic increase in variance in the population. This is a phenomenon known as decanalization. My lab works on variance a lot. Variance as a phenomena, and so this is a very interesting pattern for me. But most interestingly, in that context, you'll notice that some individual sort of remain within some homeostatic boundaries, like they don't change the phenotype very much, while other people really drift away. And you can imagine if you care about disease, there might be some threshold right here, and when you go above this threshold, you end up being sick, right? So if this is the same population, right, same individuals, and then all of a sudden just reveal a bunch of disease traits just by changing the environment, right? So this idea of decanalization increasing variance is one of the most robust observations. There was a review paper a few years ago, been looked at in like 120 some species, like under stress variance increases. It's a very, very robust observation. But the overarching question my lab is interested in is like, why do some individuals appear to be more sensitive to environmental perturbation than others? So why is that? Why are these individuals staying within these boundaries and these others drifting away from this expected norm? And if you're an evolutionary biologist, by the way, it's not just about disease, right? This actually might be an adaptive response, because maybe like these individuals are the ones who survive because they have all of a sudden have some extreme phenotype. So it's not just about disease, it's also about evolution more broadly. But one of the reasons one may care about this from a disease perspective, this will come as no surprise to people in this center, the causes of mortality in the blink of an eye from an evolution perspective have changed dramatically, right? This is in the US, but I could have shown you that for the world. We used to die of infectious disease, now we die of non-communicable disease. At the end of the day, how we understand broadly what's happening, right? We eat food that's making us sick, and we're not as active as we used to be. You could summarize this that simply. However, it is not that simple, because these conditions like obesity, type 2 diabetes, asthma, depression have a strong genetic basis. So they're rooted in the environment, but they have a strong familial correlation for these conditions. Therefore, there is a strong genetic contribution to sensitivity to this disease. Therefore, there is something to be learned in digging some genetics. But this idea about why there's such an increase, the idea that there's such an increase in the proportion of non-communicable disease has driven this obviously famous, especially here, this evolutionary mismatch hypothesis, right? This idea that we have evolved as a species under a different set of circumstances. Genetic variation in our species was shaped by a past condition, and the environment now is sort of clashing with those conditions because we are experiencing very different environments. So sorry, the genetic makeup is clashing with our environment, making us sick. And it turns out that this hypothesis has been discussed and tested in various ways, but at the genetic level, there's been very few formal tests of the evolutionary mismatch hypothesis. And this is sort of partly what I'll be telling you about today. To make sure we're on the same page, in theory, evolutionary mismatch should create genotype-by-environment interaction of the form I'm showing you here. What we have here, this is disease risk. But again, it could be any number of phenotype where there's a polymorphism, there's a AT polymorphism, where in the ancestral environment, there is no phenotypic variation, everything is good. All of a sudden, you change the environment, and this polymorphism acquires a phenotypic expression. And you see now that, because I was showing you earlier, if you have the A genotype, it makes no difference, you're fine. But if you have the TT genotype in the ancestral environment, you are fine. In the new environment, you're not so good anymore. And right, so there's sort of it creates a health disparity gradient that is stratified by genotype. So another way to frame this question is, do modern disease arise from our genomes living in the past, right? And so can we mine the past, the footprint of selection on the given genome, figure out like, oh, maybe this AT polymorphism was actually doing something, it was neutral or under positive selection, doing something beneficial in the past. The environment is not there anymore, and now it's making us sick, right? So this is sort of what we're going to be testing. I'll tell you 3 stories. Hopefully, it's not the classic vignette thing. I give you some coherence to this narrative. But I'll begin by telling you some story about fruit flies. That was work that was led by 2 postdocs in my lab, Amanda Lea and Luisa Paredes. I'll tell you a story about cell lines and a story about, indeed, the Turkana project. Okay, so we're going to start with the fly project. very briefly, but again, we can torture flies in all kind of ways that we cannot do in people. So flies are still a role to play in allowing us to test hypotheses, like the one we're talking about today. And why lifespan? Lifespan is like obviously tightly linked to fitness, especially in flies, where fly reproduce until they die. So if a fly live longer than another one, they have potential to reproduce more. So that's a perk. There's extensive genetic variation for longevity in flies. So you can actually use a very simple selection experiment and expand the lifespan of a population of fly by 70% in just a few generations of selection. We're interested in manipulating the environment, and diet is a very easy way to manipulate the environment in flies. I mean, you've all heard about caloric restriction experiment extending life. Well, in this case, we're going to use sugar as a stressor, as a new environment. We're going to give flies a 23% sugar diet, and flies experience sugar stress the same way we do. They become insulin resistant, they get increased triglyceride, they become less active. So that's going to be our stressor. Normally, the way people do lifespan study in flies, they spend a lot of time measuring the actual lifespan of individual flies using inbred line most of the time, and that's very time-consuming and painful, and I'm not that brave. So we're trying to think of a different strategy to do that. That did not involve us measuring the individual lifespan of each fly or its genotype in a population. So we have this idea of like using very large population cages. We went in some orchard, brought a bunch of flies back, put them in this giant smelly cage in Princeton. And then this is what we're going to do. So like, if just pattern 1 second for this, like the details matter here. So we take this giant cage full of flies. These flies lay a bunch of eggs, well over 100,000 eggs in small bottles. And we're going to split these eggs, which come from the same generation, into 3 cages that are going to be control and 3 cages that are going to be high sugar exposed. Okay, so now they're all age matched at the beginning of the experiment, and we're just literally going to let them die. And we're going to stop this population when only 500 flies are left in the cage. We're going to start with about 20,000 flies, and when 500 flies are left, we're going to stop the experiment. These 500 flies that are left in the cage at the end, they are our centenarian, right? They are the fly that lived the longest. And so we're not measuring lifespan for anything, we're just letting flies die. And what we're going to do is estimate the difference in the frequency between the young flies and the very old flies. You notice that high sugar is toxic to flies. They live about not quite half, but about half as long as they do in the control population. And that's it. So again, what we're going to do now is we're going to take a bunch of flies from this, at the beginning of the experiment, and we're going to compare the allele frequency at this time 0 and compare it to the end. So if an allele is beneficial between young and old, you'll see that it will increase in frequency in the population. If it's deleterious, it will decrease in frequency, right? So we're going to enrich our long-lived flies for alleles that are protective against the deleterious effect of aging poorly. This design has 2 benefits. One, we can start with a very large number of individuals. And the other one is that we can actually look at genotype-by-environment interaction indirectly, because if we find an allele that's protective, let's say, in both environments, right, they'll have the same effect in both conditions, or we can compare it across condition. Maybe there's a situation where an allele contributes to variation longevity under control condition, but not the high sugar environment, and vice versa, right? So this is like an interesting way of like looking at G/E. The problem is that we have to do a lot of sequencing. So we sequence about 10,000 individual fly in this experiment using some high throughput method, looking at a bunch of SNPs. And what do we find? Well, we find a bunch of loci. But the most interesting result that came out of this is that about 1/3 of the alleles that were involved in the variation in longevity in this experiment were context specific. So they affected variation in lifespan in one environment, but not the other. So if you only did this experiment under control condition, we would have missed about 1/3 of the allele and their contribution to lifespan. Okay, so now what we have is a pile of polymorphism that have a shared effect across environment, and a pile of polymorphism that have a context-specific effect. Okay, and now we can begin to study some of their property. Okay, same page. Excellent. So let's dive a little bit into testing this evolutionary mismatch hypothesis. So the first question we can ask is, are the detrimental alleles more likely to be evolutionarily recent, or so are they more likely to be derived or ancestral? Because we have very nice phylogeny in flies, we can polarize each allele based on whether it's ancestral or derived. And what we find is that indeed longevity decreasing alleles are more likely to be derived than ancestral. And that signal is even stronger for these alleles that are context specific, right? We have a very strong deviation compared to randomness. The next question, which is very indirect, is, are the deleterious allele that were beneficial or neutral were they beneficial or neutral in the ancestral environment? So flies are follow human out of Africa. We have more or less the same demographic history. You can follow human, you follow flies. And so we can compare, we have enough data in the fly world to look at the allele frequency pattern in Africa versus Princeton. So we're going to compare these two populations to answer this question. So the first question is, what's happening in terms of the lifespan decreasing versus lifespan decreasing alleles? And so we can see that the lifespan decreasing alleles have a lower frequency. You can see that here in the Princeton, in Princeton. Okay, so that's an interesting observation. But what happens in Africa? So again, the ideal experiment here would be to actually go to Africa and replicate this whole experiment on the African flies, right? So we can actually make a proper statement about what it looks like there, but that's the next best thing. And then we can look at the same allele, the Princeton life decreasing alleles in Africa, and we found out that they actually have higher frequency in Africa. So basically what that means is that the allele that are deleterious today in Princeton have a history of selection in Africa because they are higher frequency there. So this is again not the ideal data set for this, but like we can say that the lifespan reducing alleles that were beneficial in Africa now are paying a cost when they're in Princeton on that. So there's a very strong mismatch effect. So what have we learned so far? Well, there's a tremendous amount of G by E when we have the power to see it, right? Like a third is a very large number. The history of an allele does impact the contribution to lifespan. That's like a pretty strong signal. But the pattern that I'm most intrigued about, without being too meta, is that this is an experiment we did with one stressor, right? And we found that 1/3 of the alleles are uncovered anew. If we did this experiment in 100 different environments, right, are we going to discover that there is like more and more cryptic variation that are being unmasked that is now contributing to variation in fitness or lifespan in this context? And I think the answer would be absolutely yes. So I think, and we've actually done this experiment again using hexavalent chromium, which is a stressor flies we have never experienced, and we see a lot of variation there. So I think it's something worth keeping in mind. So this is published, and again, I'm going to try to do more of these experiments to see if we can uncover more of these allele. Okay, so continuing to try to test this evolutionary mismatch hypothesis in a different context now, moving a bit closer to humans. Of course, what we'd like to do is be able to control the environment, right? So again, we cannot do this in human, but we can do this in cell lines. So this is the work again of the wonderful Amanda Lea, who's now a faculty at Vanderbilt. Many of you, I think, know who she is. So to make sure we're on the same page, I'm showing you here an expression QTL. So all I'm saying here is that there's a given polymorphism. So maybe in the promoter of a gene, there is associated variation in expression. So if you have the 0 allele, you make less expression of that gene than if you have the 2 allele, which is like A versus TT, for example, right? So EQTL. And the question we're going to ask now is like, how does embryonal exposure impact the landscape of regulation? So can we uncover G by E effect at that level? The experimental design is really straightforward. This was an experiment we did just at the beginning of the pandemic. It was a lot of work. So we actually took cells that are LCLs, so they're B cells, they're immortalized B cells, and we're going to expose them to 10 different treatments, actually, depending how you count. And so that's a total of about 5,000 individual RNA-seq experiments. and we're using a 3' bias expression data set approach. These cells come from the 1000 Genome Project, so we have access to all the genotypes. We're going to sequence them ourselves. We have very high quality genotypes, and they come from 2 populations, from Europe and from Africa. We're curious about the effect of ancestry on this G/E signal, but that's for some other time. And the exposure we use are of 2 kinds. So we have 6 exposures that are, let's call them evolutionary familiar. These are things like hormone signal or immune stimulation. So things that cells as a B cell would have expected, we expect to have been experiencing. And then a bunch of stimulation that are like things cells should never see, like BPA, Teflon, acrylamide. And the question here will be, going forward, like if a cell has never seen this kind of stressor, do you have more or less adaptive response for it? And do you see more or less EQTLs associated with it? So what kind of pattern do you expect to see? Well, some EQTLs may be ubiquitous. So like regardless of the exposure, a given polymorphism drive variation in expression, regardless of the exposure. Some might be unique. So like one single exposure may drive expression of a given polymorphism to be an EQTL, and some may be a bit of both, right? So unique along a distribution. So what does it look like in practice? So this is one genotype, and this is one gene expression, and you can see each of the dots is an exposure, and you can see that this is a universal EQTL. So regardless of the exposure, this is an EQTL. You can see the effect across, and these are on log2 scales, so it's kind of a large effect. And here I'm showing you an example where only 2 exposures drive variation expression, the other ones do nothing, right? So this polymorphism, again here, this polymorphism is sort of, we can consider it as a cryptic polymorphism where the phenotypic effect of this allele on gene expression is only revealed under these 2 exposures, but not under these exposures. Yeah, okay, good. So what do we find? Well, 77% of the exposures drive this sort of are ubiquitous, and about 22% of the exposure drive EQTL in a sort of context-specific way with a distribution of shared effect, right? So this is how much of this is just this is unique. These are EQTL where only one exposure drives the variation, 2, 3, 4, 5, etc. So again, if you are a quantitative geneticist, especially in human, like the expectation is that genotype-by-environment interaction won't drive much variation in phenotypic trait. That's sort of like that's sort of the thinking in the field right now. But like, these kind of experiments sort of challenges this expectation because we do find a lot of genotype-by-environment interaction when the experiment is sort of designed to uncover it. One of the coolest results that came out of this paper is this. So this is heritability, and this is so again 4 gene expression, and this is what we find in our 2 control condition. And this is the sort of the expected heritability that you get. Many people have looked at heritability of gene expression in cells, and like 0.1 something, around 0.1 is what people get. But you can see that under exposure, under exposure, this heritability increases quite dramatically. And this is consistent with a bunch of data that came out from like studies in Japan, for example, where you can see the heritability for BMI has increased since the Second World War steadily. So again, like how do you explain increase in heritability? Well, G/E is like, given that the genetic makeup of this population hasn't changed, invoking G/E is probably the most likely explanation, where you mask formerly cryptic variation, you increase additive genetic variance, and all of a sudden you have an increase in heritability. I'm going to go through this quickly, but one of the question I mentioned at the beginning was this idea of like familial exposure versus non-familial exposure for the cells. And here, the first question we ask is there, so we can, because these are published data, people have, or we could have, it was available, people look at the footprint of selection for every segment in the genome, so we can ask, okay, for every EQTL, do you fall under a region that is associated with different degree of selection? So here we're using IHS as a metric for selection in the genome. And we can ask, is there something different between the context-specific EQTLs and the one that are ubiquitous? And we can see there's clearly a difference between the two, which is like interesting. But the most interesting pattern is really the fact that the evolutionary novel revealed EQTLs have a different pattern on this interval association with IHS compared to the evolutionary familiar one, where evolutionary familiar EQTL, maybe not surprisingly, are more likely to be associated with footprint of selection than the evolutionary novel one. And this is consistent with some prediction from evolutionary mismatch hypothesis, where the history of immune exposure, in this case, has driven selection for EQTL that are mobilized under stress. Okay, so again, this 22.5, 23 is a large number, right? I think like the fact that the flight data earlier and this data set are in the same direction, right? We indicate that we are missing a lot of variation, but not really considering this explicitly in many models. The history of exposure does make a difference on the pattern we see. And I didn't tell you that, but like the context-specific EQTLs are associated with disease. So this is like also an interesting factor. If you want to hear more about this, again, Amanda's paper was published in Genome Research last year, and it's available right here. Okay, any questions so far? No? Okay, excellent. Okay, so we're going to continue to test this evolutionary mismatch hypothesis in different contexts. So I'm going to dive into the Turkana work. This work, again, I don't have to talk about this in this. I don't have to go on about this in this at this center, but there's been quite a bit of work comparing subsistence level population and European population, asking questions about risk of non-communicable disease, right? So here, we can. I'm just pulling a few of them, but like we can see that the risk of type 2 diabetes is much higher in European population that's from the US, but than it is in subsistence level population. But most of the time, people that have addressed this question, again, I've been comparing subsistence level population to population that live either in Europe or in the US, right? And rarely, and so the genetic makeup of this population is different. The timing of transition is very different. So we're interested in finding a population where we could have this gradient of lifestyle within a genetic background that was more or less uniform. And that's what I'm going to tell you about today. So this takes us to Turkana, which is a region of northern Kenya. So this is South Sudan, Uganda, and Ethiopia is right here. And this story sort of had a maybe unfortunate origin. So actually a few years ago in 2018 or something like that, I lost all the flies in my lab. Like everything died basically. I was a young assistant professor and I lost literally everything, so malfunction in the building. So to sort of get over myself, I went to visit a friend of mine who worked at the Turkana Basin Institute in northern Kenya, TBI. And we were taking a hike in a place that looked just like that. These are the Napadet Hills, if some of you know Turkana. And I came across 3 women who were carrying water on their head, maybe in the front and the back. They were must be like 16, 18 at the most. And asked my friend, like, what are they doing? Because like, there's no housing anywhere in sight. And he told me, oh, these are ladies from the Turkana community, and they walked 3 to 6 miles to get water back to their home. And I walked back to the car completely exhausted and dehydrated, wondering how in the world are they doing this? Maybe that's what I'll do for a bit. I'm going to come back here, see they're interested in working with me. I'm going to study desert living. I'm going to figure this out because that's the best I could think of at the time. And indeed, Turkana is a very hot place. It barely rains. And when it rains, it's very sporadic. We get 2 rains a year, and they used to because now with climate change is even less predictable. And it's very hot. So these are average temperatures. But temperature getting, I guess you get used to this here. But 120 is not unusual. And so yeah, I went back to Kenya, lived there for a bit, and engage with various people in various communities and see if they were interested in like partnering with us to study this problem. And that's how I got to know the Turkana community. And I confirm indeed that water scarcity was really a driving force shaping the lifestyle of this population in so many ways, whether it is to feed, to get water for the animals or for themselves. whether it's like they're a semi-nomadic pastoralist community, so they rely, I'll tell you about that in a second, but they rely on the livestock for survivorship. And so even the rhythm of where they move with the animals is governed by water. But what I discovered as I was, again, engaging this community to sort of figure out what, if we could work with them, and they were interested in the question we were asking, is actually their recent history. So up until late 70s, the exact date is like fuzzy because I think it wasn't uniform, but there was a military blockade of the northern part of Kenya for quite a long time. So the British had it in place and the Kenyan government kept it up for a while after the British left. And basically what that created is a very dramatic difference in lifestyle, people that were living on this side versus this side, where most population living north of that boundary were mostly stayed away from market economies up until this was removed. And then more dramatically, again, this is like a very clear example of like the community driving the question that we're now asking, because I would have never thought of this or gone down that path if it wasn't for some elders telling me, what's most dramatic about our life is like, I was born in that time, and like afterwards things have changed so dramatically. And so now what we have is a situation where their population living sort of really in far remote corners where they're still living today a non-market lifestyle, where they live very traditional subsistence level lifestyle, and population that have embraced market economies for better or for worse. But the most dramatic event, there was actually a famine that some of you may remember in the 1980s, like 1981, 2, 3, and it was the period again is a bit fuzzy here, where like many people had to move to food aid or immigrate out of the Turkana basin. So this sort of formed the basis for the gradient that we're now studying. So a traditional Turkana person, 80% of their diet rely on animal byproduct. That's mostly milk, blood, and meat, and then real milk being the main staple, and then a little bit of grain and a few other things they find in the environment. And they're also very active. So we put podometers on some participants, and like I was shocked to find out how much they walk. I mean, they're really very active. I don't know if you've checked your podometers recently, but you're probably very far from there. And so the framework here is that we have people that live traditional lifestyle and people that live more urban and integrated lifestyle. And one of the tenet of the mismatch hypothesis that there's some disparity across environment in health risk outcome. So the very first project we undertook is like establishing the fact that there are health differences between traditional community and an urbanized community, or at least market integrated. So there was a lot of community engagement, a lot of conversation with various stakeholders and partners, and we started this project in 2019. And again, the main focus was initially on like establishing the health consequences of this gradient. So we measure a lot of things. So we give people questionnaires. We partner with various anthropologists to establish this questionnaire. We measure as many biomarkers as we can. The list is like, this is an example. We have almost 500 phenotype per participant in the database, and we collect a lot of samples. So we have, I'm going to tell you about it in a second, so it matters here. We collect blood for DNA. We measure, we do white blood cell count. We have PBMC, which is the immune system. We collect serum to measure a bunch of biomarkers and do metabolomics. We have urine samples, which I'll tell you about again, and we have fecal sample from microbiome. We also have like nails and like a bunch of other things. So the first paper to come out of this project, which again was led by Amanda, was a paper where we established that there's actually strong health gradient between traditional communities and people that are market integrated. Especially here, we focus on cardiovascular health. Interesting, we find some nonlinear pattern between the traditional population that live in Turkana, people that are living rural lifestyle in Turkana versus people that are living urban lifestyle. But like there's definitely a strong health gradient that we can now exploit in a way to study a bunch of correlates that go alongside this health gradient. But the real first step that sort of motivated part of this project is establishing that the history of selection on the Turkana genome is associated or not with now the emergence of non-communicable disease. Okay, so that now is taking a step back, and we are going to go back to asking this question that I thought I was going to focus on initially, which was like this question of arid living. So we sequence a number of Turkana. We actually, with the help of a group from here, which I'll talk about at the very end. So we have a total of about 301 Turkana samples. And we were very concerned that because of the part of the lifestyle, there's also a raiding community. There may be a lot of admixture with neighboring communities. So we also sequence the genome of community that the Turkana interact with on a regular basis. Again, the expectation there will be some admixture between the two. It turned out we didn't find any, but and so now we can look at two patterns of selection. One is sort of the classic sweep pattern that you are probably familiar with, and the other one is a pattern of polygenic selection, where there's a more diffuse signature. I won't have time to talk about that today, but I'll focus on the sweep, on the hard sweep. When I started doing this, I never quite realized how a trickier game this was, because we can measure various statistics that are associated with the footprint of selection, but there's no proper null. So there's no proper statistical test that one can use to say this allele is under selection versus not with the actual p-value. So what we can do is like draw a distribution of statistics and ask whether or not we're in the tail of the distribution, whether that tail is fatter than you expect by chance. And so to avoid the traps of like, because every tail is a distribution, we actually look at different statistics and look at the intercept across them, because they rely on very different assumptions. So we use a PBS, which is a population branch test, and we use a Luya and the Eurubens, which have data available, publicly available, to compare to Turkana. I was, again, this is like, my background is in flight genetics, and I was shocked to see how, I mean, not anymore, but like how few African, especially East African genomes are publicly available to make this kind of comparison. They are very, very few. Like this Turkana data set is the largest single cohort that we generated through this teeny project in a way, that are going to be eventually publicly available. And so we use the PBS test, the IHS test, which is a test of X-linked haplotype, and then the XTX test, which also look for selection accounting for population structure. And so what did we find? We found a few really strong outliers that were common across these tests. So here I'm just talking about this pile right here. And a couple of them had been described previously, so they're known to be under selection. But the main signal came from this gene called STC1. It was a very strong outlier. We were sort of happily surprised to see that. It was got lucky, I think, because STC1 actually has been well described. It's a very well studied gene, and it's involved in glomerular filtration rate, which is how well your kidney are filtering your blood. It's also involved in urea levels. So remember the Turkana primarily eat meat, right? There's a huge amount. Their protein intake exceeds by 300% the World Health Organization recommendations. They have a very high protein diet. This gene was also implicated for GWAS in a bunch of biobanks in chronic kidney disease, and again, creatine level, which ties back to urea. So that was like, oh, that makes sense, right? So I was just getting ready to contact some friends of mine to do some mice experiment, to dehydrate them and see what happened, and like just poke the gene, right? That's what you would do. But we were lucky because again, someone had done the work for us. So there are two papers. One of them that showed that this STC1 gene is upregulated in rats by 8-fold under dehydration stress. So it's really responsive to dehydration stress. And another experiment that showed very convincingly that this gene is actually regulated by the antidiuretic hormone. So when people start sweating and getting, you know how this works, this gene also is upregulated. Something that we also validated ourselves in the lab. The last little snippet I'll tell you about that gene is that we work in partnership with Ian Vasconcello, who was a grad student in Philip Messer's lab at Cornell, and he did some. I have to say, like I can do some PubGen, but this is like this is magical. used some AI algorithm to time the onset of selection in this population to about 350 generation. And he used different approaches to do that, and they both converge on the same answer. And not only that, but also he estimated the strength of selection to be really quite, I mean, this is a very large selection coefficient, 0.04. It's in line with like some of the stronger selection we have, and the Phi allele, for example, of malaria, or some of the most popular examples. But this 350 generation is also interesting because if you, depending on how many years you can compute per generation, it's also the onset of the end of the African arid period, which is also when pastoralism as a lifestyle evolved in humans. So when this Nilote population, say like moving away from the Nile, and so 5 to 7,000 years is roughly when that should have happened, which is roughly what we find. And I'm sure some of you here know much more about that, so I will leave it at that. But it's cool that the timing works out. And the last little story I tell you is that we, just to go back to this evolutionary mismatch idea, we actually took PBMCs, I was telling you, from some of these participants that live across lifestyles, from rural and urban population. And we see that there are a number of genes that are differentially expressed between them, with surprising a very strong bias toward higher expression in the rural area. We're still trying to make sense of that, out of that bias, because that's really, really strong. But no matter what we do, the pattern doesn't go away, so it must be true. And then we then ask of that pile of 500 some differentially expressed gene, which one are known to be associated with disease or specific traits. So there are databases in humans of disease traits. It's called, this is the reference for the disease database. And we find a strong enrichment for cardiovascular disease and type 2 diabetes in the differentially expressed gene, which again is driven by the difference between urban and rural area. And the thing we can next then do is ask for these genes that are associated with disease versus ones that are not. Are there SNPs that are more likely to be involved in selection or not, right? So we can ask, is there a history of selection associated with the gene that are differentially expressed, and which direction does it go towards? And we find indeed that there is a strong effect where the gene that are associated with disease are more likely to be associated with the history of selection in the Turkana, which is cool because it again fits that prediction from the evolutionary mismatch hypothesis. So in conclusion, we've identified the genomic signature of adaptation to arid living in humans. I was surprised, but as far as I know, that's like the first time people actually look at that explicitly. I didn't tell you that, I skipped that, but just I should mention it. There's strong evidence of polygenic selection associated with renal function. And so the same pathway we find from the heart sweep is also sort of recapitulated in the polygenic sense. And the SNP that fall upstream of genes that are differentially expressed by lifestyle also exhibit evidence of selection, more so than genes that are not associated with genes that are differentially expressed. Okay, this paper is on bioRxiv. It's under review. Hopefully we'll hear soon. We still haven't heard, but we should hear soon. A huge thank you to the participants. I came into this project, again, as a fly geneticist, learning a lot about how to interact with communities. Like I really had to do homework from scratch on this. And again, Amanda was an amazing partner to do that with. But I was very happily surprised to see how willing and engaged the communities we work with are. They are very interested in doing research with us, and that has been very heartwarming. Along the way, what I came to appreciate, something again, Amanda and I have been like really thinking about a lot. There are many people, including people working here, that are working with subsistence level groups around the world, so we have a very similar project in my lab in the South Pacific, in a country called Kiribati, where we have islands that are really remote, where people have much harder access to markets, where there's another set of population that live in the main island, and the same health disparity that we see in the Turkana manifests itself. And again, many other projects, the Chimani, the Shuar, the project in Madagascar, I mean, there's so many projects around the world. People in anthropology have been comparing health disparities across traditional communities and population transitioning to market integration. And the metaphors are the same health, the same disease, the same social consequences. And I think given how fast populations are transitioning away from sustainable lifestyle, we have a very short window to address these health disparities and understand to what extent these populations are going to be suffering disproportionately from our so-called diseases of civilization. So we got a group of people together and wrote this perspective article, which hopefully will come out soon in PLOS Biology as an essay, advocating for a greater focus on the health outcome of this transition and more work in partnership with sustainable population. Okay, so take home, I'm done. So here we set to shed some light on this evolution mismatch hypothesis from a genomic perspective, and we're just getting started. So hopefully more to come on that front. We hopefully I try to convince you that when we have the right design and enough statistical power, we can identify a lot of G/E signal. I think the field needs to wake up to the reality of the importance of context dependency in genetics. Along those lines, this idea of cryptic genetic variation, which is so popular in model system, hasn't quite made its way through human genetics. So Greg Gibson was one of the pioneer thinking and describing that pattern. He wrote a beautiful book, by the way, about that called something about the clash between the genome and the environment. Greg Gibson, you should look it up. It's a great book. And now we have the tool, and that sequencing is so cheap and easy to identify the footprint of selection and test at the molecular level, at the genetic level, key predictions from the evolution mismatch hypothesis. I'm done. I welcome your question. This was great work done in partnership, as I was mentioning, especially for the Turkana. The STC1 story was really a partnership with people from here. So again, thank you. I have great collaborators in Kenya and an incredible team of now 14 people who work all the time in Kenya, and without which this work couldn't be done. Thank you for your time. Any questions?
Great talk, Julien. That's
Thank
really
you.
cool stuff.
Thank you.
I was especially excited by the STC1 paper. I can't wait to read that. I was just wondering, you showed that there were like 4 or 5 other genes that also showed signatures of selection. Do you know what the story are for any of those yet?
We have many others. We have not drilled down. I mean, like, so some of them are involved in, I'm gonna say endurance, broadly speaking. So there's like assuming more like lung capacity, heart size. I mean, it's a long list, right? So yeah, we're looking at them. So assuming metabolism quite a bit. I mean, there are so many remarkable things about the biology of the Turkanas. Like one of the projects we are now going after is cholesterol. So a very unusual cholesterol pattern. They have some of the highest HDL of any population in the world, which is like really unexpected. Like Inuits are the closest to rank. So there's some really remarkable patterns. So I suspect some of these genes are involved in that as well. But like I'm speculating here. I don't know.
Have you looked at lipoprotein little a?
We are measuring it now. Yeah. I have very high LPA, so I'm like very obsessed with it. So yes, like, yeah. Somehow in the 90s, like LPA was measured routinely in the US also, because it's like the single best predictor of like adverse cardiovascular outcome. But then people realized they can do nothing about it because it's like purely genetic. And so they stopped measuring it. So like I'd be surprised if any of you get LPA measure when you get a physical, because even if it's really high, nothing you can do. It's in the clinical trial. Yeah. I'm counting on it. So yeah.
Cool. I have a question about the last bit on the selection you saw in the Turkana population. You showed the one allele, right? Reaching the simulations were about one allele, right?
Yeah.
Well, going from 5% to like 80%.
Yeah. Wanted to pick a SNP in that window.
Do you have any idea what that selected allele like directionally does, right? Like what does it do to the gene in any sense?
We don't know, unfortunately. So we don't actually. So all these SNPs are in like a big window of like about 70 kb, And sorry, all the SNPs are, and we don't know which polymorphism is actually causal.
Is it all linked?
So they're all linked. So we picked a SNP that sort of tagged the haplotype, and then so the simulation is on that tagging SNP, which is one of the many we could have picked. So unfortunately, so it's upstream. So we know it's regulatory. I skipped that. Right? We know that window is upstream of STC1. We know it's regulatory for STC1 with some contact map that shows that convincingly. But like that window, yeah. No, a subset of the window, but it's a big window, yeah.
Cool. We got a couple questions online here.
Yeah.
One's from Mike Irvin.
Oh. I tremble. No.
Yeah, you're getting.
I know. Please.
Any insights into why Turkana have higher than expected hypertension, including those living the pastoralist lifestyle?
No, we do not. That is a good question, but that is a great question. No, there are a lot of like we are not yet, the project isn't mature enough, I think, to the point where we're getting there, right? We have about 3,700 some people that have been enrolled so far, and we have measured a lot of them, but like to get to this kind of like mechanistic underpinning will take quite a bit more work. Yeah, Mike, if you can help us figure this out, please.
I have another question, but then I'm going to shift to flies really briefly. And then, it is the context dependence is super important, right? But then you also showed that heritability increased, right, in under context, under certain contexts, right, after stimulation. And you said there was like an increase in the additive genetic variance, but
that's the hypothesis, but yeah.
Could it also be a decrease? Like, why is it not a decrease in the environmental variation or in the total variation, right, overall?
Because in general, we don't see that so under stress variance tend to increase, right? So I mean, it could be like, yeah, it could be both, right? It's not, I don't think it's that straightforward. I mean, yeah, I simplified it a bit, but I think the most parsimonious explanation to explain the increase in heritability is indeed that you have mass cryptic variation. It's like some polymorphism are prone to blink out. So I'm saying that some blink in, some will blink out as well, right? That's also going to happen, much less from what we see. But yes, you cannot discount the fact that the overall phenotypic distribution changes, and that in itself can change heritability. That's your point, right?
Yeah, like if under stimulation, do they all hit some ceiling, right?
Yep.
And then your total variation is smaller, right? In general. Yeah.
But that's not what we see.
So I was wondering whether you're seeing this STC1 in outgroup populations that are not pastoral. And a related question, which is that if the selective sweep is like almost 4,000, 5,000 years ago, then it's partly associated with the entire like Nilo-Saharan complex, which means it should be really widespread and not just unique to the Turkana. So you did have some populations, Nilo-Saharan populations like Karamojong, Maasai, etc. What do you think is happening there? And also, I guess, if you don't have this outgroup where you don't know that, there's in the selective sweep, how do we know it's like, part of the arid adaptation?
Absolutely. So this is a very good question. You'll be good with review number three. It's like spot on. So we actually don't know. So first of all, this is not de novo mutation that just appeared in the Turkana. It is clearly segregating in Africa in most other populations. So that's an important point. I mean, I didn't want to get into the details, but like that's critical. I would expect exactly that it would be shared between all these members of the all the Nile nodes should have the same sort of pattern. So actually, we're testing that now explicitly, even with the Kushinic. So Amanda and I are very curious about this. So we just sampled 300 some people from different communities, like with enough number. We are like 7 or so. It's not quite enough what we have. So we sample more people from these different communities. I think we picked 3 Nilotes and 3 Kushinic populations to see, one, is it the same signal across them? And 2, like what is the frequency across these populations? Because my expectation is just like yours, like is that it should be common shared across all of them. And if it is not, we have to reassess our hypothesis. Yeah. I mean, but clearly there's something very unusual about that gene. So like it is like the frequency change is very dramatic, right? And is without a doubt under selection. The question is like, what is, yeah. So we have other evidence that I didn't present here that are.
Yeah, I mean, just one interesting thing is that the Turkana living in literally the driest habitat of all of the Nilo-Saharan groups, and they're by and large, like many of the Nilo-Saharan groups, are living in not too, not too bad, too dry, like they're mostly agro-pastoralists. So I guess one possibility is that the magic stats that happened there is kind of like pushing the date more back than it might be, but or it could, like, I wonder whether you think that maybe there's an early origin, but that in the Turkana there's been much greater persistence of that trait because of their super arid environment.
Yeah, I think that the latter is probably the most likely. I mean, again, it's purely speculative, right? The demographic history of this population is very poorly defined, right? Like, even how long have they been in the Turkana basin? So clearly they have not been in the Turkana basin for that long, 400-500 years maybe. Before that, they experienced environment that were very similar in northern Uganda and right, all the way up there. But like, so like they've been living under arid condition for a very long time, for much more than they have been in the Turkana basin. But like, what happened? Like, what was the environment going back in time? Where were they? What path they took? None of this is known. So, I mean, that's one of the things I hope we can, like, by having more genome for more communities around, like, I mean, also one of the surprising results is that, so we saw no admixture, but like also the Karamajan, the Turkana, some of these communities, like, we can barely tell them apart in terms of biological frequency differences. It must be a very recent split, right? So, yeah, there's a lot to be described in that context that is above my pay grade. But I don't do that kind of population genetics. Yeah. Thank you so much. Yeah. Okay.